Concept Map for Biology 2440. This concept map is over membranes, and here at the center I put membrane proteins. And membrane proteins are where all the functions take place. They carry out the functions of the membrane, and that is the plasma membrane that I'm talking about. It is on every cell, and it is made up by a phospholipid bilayer, which is right here. And the phospholipid bilayer is made up of amphipathic molecules. It's double layered, and it has they have hydrophilic heads and hydrophobic tails. Hydrophilic is water loving and hydrophobic is water repelling. And hydrophobic tails can either be saturated fatty acids or unsaturated fatty acids, and the difference is saturated have as many hydrogen bonds as possible, and unsaturated have double bonds, not leaving room for hydrogen. And I put glycolipids onto hydrophobic tails. They are lipid molecules that have a carbohydrate group. And going back to the phospholipid bilayer, detergent molecules is coming off of that. And detergent molecules are also amphipathic, and they break up the membrane. They break up the phospholipid bilayer. And coming off of plasma membrane is also selective permeability, and this means that the membrane can choose what to go through and what not to go through it. And something that can go through it is water, and when water diffuses through the membrane, it's called osmosis. And water will go passively from the more concentrated area to the less concentrated area to equal out the concentrations. It will go from the less concentrated area to the more concentrated area. And selective permeability, coming off of that, is simple diffusion, and simple diffusion is kind of what I was already talking about with water, except it will go with molecules, and it's just when things can go through the membrane freely. And what I was talking about with water is the concentration gradient, and this is when the outside of the cell is a different concentration than the inside, or the inside is a different concentration than the outside, and they can even be the same. So when there's equal amounts of ions or other molecules on either side, the solution is isotonic. And when there's more on the outside than the inside, it's hypertonic. When there's a higher concentration of ions or other molecules on the outside than the inside, it's hypotonic. And that will dictate whether simple diffusion and active diffusion, which I'll talk about later, allows things to go through. And it connected to the concentration gradient is the electrochemical gradient. And the electrochemical gradient is based off of chemicals like ions, like the concentration gradient, but it's also based off of the electrical charge on those ions. So chlorine has a negative charge on it, sodium has a positive charge, and connected to that is protoglycan, which is what makes up the outside of between cells. And then going back to membrane proteins, and we'll go to integral and peripheral. Integral is permanently connected to the membrane, and peripheral is going to be easier to take apart without ruining the membrane. Connected to integrals, transporter proteins, and these are proteins that help with diffusion across, or permeability across the membrane. And there's active or passive transport, and active uses ATP energy and passive does not. There's channel-mediated passive support, which is when the membrane proteins allow for passive transport. And there's membrane channels, which also allow for passive transport. Vesicles are when parts of the membrane basically like wrap around particles to bring them in or out of the cell, and it like disconnects from the rest of the membrane. And then we have ion transport, and that's when membrane proteins transport ions, and either through an ion channel or a gated ion channel. Gated opens and closes based off of signals that it receives. And then back to membrane proteins, 3 other types besides transport are receptor, anchor, and enzyme. We have monolayer associated proteins, and that's when the protein is only in one of the double layered phospholipid bilayer. And we have lipid linked, which means they are associated with only the inside of the membrane, but mostly they're on the outside. Just like one little part of the protein is linked. And then there's the protein attached, and those proteins are only there to attach to other proteins in the membrane. They're not actually attached to the membrane at all. And then I connected the fluid mosaic model to these 3 because the fluid mosaic model is proteins that can go all the way through the membrane versus the sandwich model, which is proteins that are only on the outside, but they're all the way along each outside layer. And so that was proven to be incorrect. And one last thing on membrane proteins, I also put glycoproteins like glycolipids, except they're proteins with a carbohydrate. And also down here are sterols, and sterols are steroid alcohols that are in the plasma membrane, and they're cholesterol.